Genetic counseling for Parkinson's disease. As part of the genetic testing portion of the Parkinson's Progression Markers Initiative, also known as the PPMI study, you will have the opportunity to speak with a genetic counselor about your test results. Genetic counseling is an educational process that includes helping people understand genetic test results and familial risk for disease. A genetic counselor is a healthcare professional with specialized training in medical genetics and counseling. One of our genetic counselors will review your results together by phone, discuss your family history in the context of your test results, review information about Parkinson's disease, and discuss the PPMI study. You enrolled in our study possibly due to a family member or yourself having Parkinson's disease, or a family member having a specific gene mutation. Parkinson's disease is a neurodegenerative disease that results from loss of dopamine-producing neurons in the part of the brain called the substantia nigra. The resultant dopamine deficiency leads to the characteristic features observed in Parkinson's disease: bradykinesia or slow movement, rigidity of the muscles, tremor, and gait impairment. Parkinson's disease may be associated with various non-motor symptoms, such as an altered ability to smell, cognitive and psychiatric symptoms, sleep disorders, pain, and fatigue. Some of the non-motor features can precede observable motor dysfunction by more than a decade. It is important to remember that there can be a broad range of symptoms, even among family members. Currently, Parkinson's disease is primarily managed by symptomatic treatment with drugs that replace or mimic the effects of dopamine. Parkinson's disease was previously thought to be caused primarily by environmental factors. Research now indicates that the disease develops from a complicated interplay of genetics and environment. A family history of Parkinson's disease is associated with an increased risk of developing it, suggesting that the contribution of genetics to Parkinson's disease may be more significant than first realized. Specific gene discoveries, such as mutations in the SNCA gene and others, have also highlighted the role of genes in contributing to Parkinson's disease risk. Research suggests that genetic factors influence Parkinson's disease risk in variable ways. Some genetic factors appear to contribute more to Parkinson's disease risk than others, representing a risk continuum. In addition, known and unknown genetic and environmental factors, as well as age, may play a role in determining when a disease is expressed. This combination of factors required for Parkinson's disease to manifest is termed disease threshold. A gene is a section of DNA that tells the body how to make a certain protein. Each gene is like an instruction, which the body reads and interprets. If there is a change in a gene, also called a mutation or variant, then this can alter the function of the gene, and therefore the protein for which it codes. Some mutations are more severe than others, resulting in different effects on the protein. We have 2 copies of almost every gene, one inherited from our mother and one from our father. Genetic diseases can be inherited in different ways, including autosomal recessive or dominant. Some conditions, such as Gaucher disease caused by a mutation in the GBA gene, are inherited in an autosomal recessive pattern. This means that a person has to inherit 2 mutations, one from each parent, to have the disease. Individuals with only 1 mutation are called carriers and typically will not have Gaucher disease. Other conditions are inherited in an autosomal dominant pattern, meaning it is only necessary to have 1 mutation in 1 gene to have the associated disease or predisposition to disease. The mutation is often inherited through multiple generations, with each carrier parent having a 50% chance of passing on the mutation to each child. The Parkinson's disease gene mutations in this study are inherited in an autosomal dominant manner. However, many people who carry these mutations will never develop Parkinson's disease. These mutations are often referred to as predisposition mutations or genetic risk factors associated with Parkinson's disease. Penetrance is a term used to describe how often a disease is actually expressed in a person who carries a gene mutation. Factors affecting penetrance include other modifying genes, age, environmental factors, and unknown factors. All of these things may determine if a carrier will develop the condition. Again, this means that not everyone with an altered gene develops Parkinson's disease. The genetic testing done through the PPMI study is targeted DNA analysis that looks for specific gene changes, mutations, in each of the genes called LRRK2 and GBA. This targeted testing will not detect other mutations in these genes, nor will it detect mutations in any other genes. As mentioned, the genetics of Parkinson's disease is complex, and there are many genetic changes that can influence the risk of Parkinson's disease. As a result, a negative or normal result does not rule out Parkinson's disease, and there is a possibility of other risk factors contributing to the risk of Parkinson's disease that may be present in you and your family. LRRK2 mutations, especially the G2019S mutation, are the most frequent contributing factors to Parkinson's disease worldwide. The G2019S mutation is especially prevalent in individuals with Parkinson's disease who are of Ashkenazi Jewish ancestry. Mutations in the GBA gene are also important genetic risk factors for Parkinson's disease, especially among those of Ashkenazi Jewish ancestry. So far, about 300 different mutations have been identified in the GBA gene, the most common of which is the specific N370S mutation. Among individuals of Ashkenazi Jewish ancestry, about 1 in 18 individuals carry GBA mutations, and N370S is the most frequently occurring GBA mutation. The carrier frequency in other ethnic groups is much lower, with a large range of GBA mutations reported. GBA mutations are also associated with a metabolic condition known as Gaucher disease. Gaucher disease is recessively inherited and occurs in individuals who have a mutation in both copies of the GBA gene, typically inherited from both parents. Individuals who are carriers of only one GBA mutation will not develop Gaucher disease. Mutations in the GBA gene lead to a deficiency of the protein or enzyme beta-glucocerebrosidase. Due to the faulty enzyme, glucocerebroside accumulates in the brain, liver, spleen, lungs, and bone, causing the organs not to function properly. Symptoms of Gaucher disease may include fatigue, anemia, easy bruising and bleeding, severe bone pain, fractures, and an enlarged spleen. Gaucher disease is categorized into 3 different types, depending on the severity of the condition and other features. The GBA N370S mutation is associated with the mildest form of Gaucher disease, type 1. It can be so mild that some individuals may not realize they have the condition. The PPMI study is a landmark observational study sponsored by the Michael J. Fox Foundation and industry partners to identify biomarkers of Parkinson's disease. A Parkinson's disease biomarker is an objectively measurable biological characteristic that may correlate with Parkinson's disease diagnosis or Parkinson's disease progression. The lack of Parkinson's disease-related biomarkers is a major barrier to the development of better treatments. PPMI is currently studying the connection between Parkinson's disease and having a mutation in either the LRRK2 or GBA genes. Carriers of LRRK2 or GBA mutations qualify to participate in the clinical phase of the study. In this phase, individuals are evaluated by imaging, laboratory, and clinical testing at a PPMI clinical study site in an attempt to identify potential Parkinson's disease biomarkers. In summary, the risk of developing Parkinson's disease is no longer viewed as primarily due to environmental factors. Instead, in most cases, Parkinson's disease seems to result from a complicated interplay of genetic and environmental factors, probably impacting multiple molecular pathways. LRRK2 and GBA predisposition mutations are important players in Parkinson's disease-associated risk. Individuals and families with these mutations may hold the clue in identifying biomarkers for Parkinson's disease, which will potentially open up the door for better and earlier treatments. In your genetic counseling session, you will have time to explore these topics in more detail. In addition, any concerns or questions relating to your genetic testing will be addressed. We thank you for your participation thus far in the PPMI study.
PPMI could answer questions that would transform the lives of people with Parkinson's. Hundreds of partners, studied volunteers, scientists, clinicians, and donors have come together to make PPMI a reality. With your help, we are seeing results that could lead to the discovery of PD biomarkers. Thank you for being part of the answer.